Good afternoon. I'm Chris O'Connor. I'm Principal Investigator of the Heart Failure Collaboratory, and we're here with Heart of the Matter, Heart Failure Collaboratory podcast to discuss the very important and dynamic area in clinical trials of representation and diversity. And I'm here with a distinguished panel to comment on this over the next 30 minutes to see if we can provide insight, a deeper understanding, and help guide clinical trials so that they can be of the highest value going forward. And so let me go around the room and ask you to introduce yourself, Dr. Batchelor.
Yes, hi, I'm Wayne Batchelor. I'm the president of the medicine service line at the Innova Health System. I happen to also be an interventional cardiologist and a clinical trialist really interested in this particular topic. So really pleased to be here, Chris. Thanks for inviting me.
Thank you, Wayne. Dr. Christian Guilliod.
Hey, I'm Christian Guilliod. I'm a resident at University of Miami, Jackson Memorial Hospital, and I've been working with the Heart Failure Collaboratory for a year in the representative population subgroup, and it's been a real pleasure, and it's a pleasure to be here.
Thank you. Dr. Orly Vardeny.
Hi, I'm Orly Vardeny, and I'm professor of medicine at the University of Minnesota, and I've been working with Dr. Fiuzat and Dr. O'Connor in the Heart Failure Collaboratory and co-chair the representative populations subgroup along with Vanessa Bloomer from Innova as well. Pleased to be here.
Thank you. Dr. Mona Fiuzat.
Hi, everybody. I'm Mona Fiuzat. I'm an associate professor of medicine at Duke University, and I'm also part of the working group that has been referred to the representative populations working group.
Thank you. Thank you for being here. The collaboratory's mission is to improve the value of clinical trial conduct so that we can get the highest quality, economically reasonable costs, but get a help the development process, the ecosystem of development of drugs, devices, biologics in a more favorable fashion, with all the stakeholders around the table contributing to how this should be changed, how we can do better. And one of the areas that we've been tackling for years is representativeness and representation in clinical trials. And I'm going to start with Dr. Batchelor to help us, because we're in a dynamic world right now where we're talking about diversity and representativeness, and the terms matter, the language matters, and yet we're trying to get to an endpoint that is better than we have been in the past and more meaningful as we go forward. Wayne, help us through the nomenclature and what we're trying to achieve in clinical trials.
Well, thanks, Chris, and thanks for putting on this podcast, because I think this is exceedingly important. There have been a lot of changes over the last few years on the nature of this conversation, and I think definitions really matter at the outset of embarking on this kind of conversation. So we have diversity and we have representation. At the end of the day, I firmly believe that we should be really thinking about the regulatory science and what matters to patients and to the data. Diversity is just simply the definition of diversity, when you think of it, is just establishing different participant groups in the clinical trial. So having some different racial groups, ethnic groups, obviously sex, age groups, and perhaps rurality versus urban groups represented. But that's not the same as representation. I think it's extremely important that we understand that representation is what we are trying to achieve in clinical trials, not purely diversity. Representation speaks to the composition of trial participants and how closely those representative patient cohorts that are enrolled in clinical trials align and reflect the intended use population. So in other words, if I'm doing a trial in hypertension, I might have a diverse group that is represented maybe by 5 to 7% African Americans, 5 to 7% Hispanic patients, but that's not necessarily the same as representation, because well over 30% of our population of hypertensive patients in the United States self-identify as being Black. It has a disproportionate prevalence. So many have many have referred to this participant to prevalence ratio, where you look at any particular demographic group and you look at the clinical trial, how well that group is represented in the clinical trial relative to what's the prevalence or the representation of that particular demographic group in that particular disease state. So I think it's really important to understand the difference between diversity and representation. And what we should probably be speaking about is representation more, which really is how well does a clinical trial reflect the intended use population? Because that's where we're getting all of our information on safety and efficacy for a drug or device. And that should be really underpinned by adequate representation in the representative patient cohort included in the trial.
And Wayne, that is so well said. And I think that's really where we want to get. And if you think about the history of where we've been in clinical trials, we've often had these mufflers or these restrictions on inclusion exclusion criteria. And so that you and I know, when we did trials in myocardial infarction or heart failure, we would end up at the end of the day with an average age that was well under what we would anticipate the intended population or the population at hand that is fully at risk for that disease state. And so one area that I think have need improvement is in the area of lifespan representation, and particularly taking down the barriers of saying, well, you can't get into clinical trial if you're over the age of 75. We've done better. I think one of the areas that I was impressed to see, Wayne, in the trials you've been involved with are the TAVR trials. The average age of the TAVR trials really do represent the intended population. Tell us a little bit about that.
Well, it's a great comment. For a while in the first series of patients enrolled in certainly the early trials of TAVR, they were over age 80. This was by design because, as you know, the first indication for transcatheter aortic valve replacement was in those patients who deemed to be at prohibitive or high risk of surgery. So age was a predominant feature in those patients. Still today, though, if you look at the average age of patients undergoing these commercially available procedures, they're still about age 80. So we're still seeing the devices applied quite commonly in older adults, and that also speaks to some of the guidelines that use age as ways of distinguishing between different levels of evidence for supporting either surgery or transcatheter aortic valve replacement in our guidelines. So there are some things that are baked into this that relate to how we define the appropriate use of transcatheter aortic valve replacement and how it actually came into play. It really came into commercial use because of the need to come up with a solution in our older patient population. So some of that's by design and by the need to actually offer a unique solution that's lower risk and less invasive for our older patients. But having said that, I'm a firm believer that we've got to have age representation in our trials more uniformly. You know, the way that we metabolize drugs changes with age. Our likelihood of having a risk of a bleed or a vascular complication is remarkably dependent on age, and by the way, also on sex. We recognize in the early TAVR years that women had about a double risk of vascular complications and were more likely to have a serious bleed or vascular complications from these procedures. So if we don't have adequate representation of older adults and women in some of these transcatheter therapies, we're grossly underrepresenting or underappreciating some of the safety signals and certainly efficacy as well in these trials. We'll miss the mark.
Well, and getting to the point of representation, Orly and Christian, you've been working on a project looking at publicly available NIH trials to see if we can develop a representation index, an indice, and Dr. Bloomer's been involved with this, and Dr. Fiuzat, taking the population of that disease state and then seeing how did the different sites and geographies around the United States perform in recruiting patients that reflect the population at risk. Tell us a little bit about that, Orly.
I'm actually going to let Christian tell us about it because he's been intimately involved and knows the data much better and can speak to it better than I could. So please go ahead.
Christian, start us off.
Yeah, definitely. So, well, I've had actually the privilege of training outside of the US also. And I think this is an important discussion because when I used to train outside, the question about clinical trials when we were discussing results was often, well, do these results matter if it doesn't adequately represent the population that we're treating here? And it took me by surprise when I came to the US that we were also asking the question, and I'm training in Miami, but we were also asking the question of did these trials adequately represent the population that we're treating in Miami? And so that's sort of the question that we had at the Heart Failure Collaboratory when we were trying to initially study if there were regional differences in enrollment of patients for heart failure clinical trials. And we've seen in abstracts that we've published that those differences do exist. And of course, the question after that was, well, there might be regional variations just because the population of those trial sites are also diverse and different from like between regions. And so then we came up with the question of how can we standardize or how can we find a way through participation to prevalence ratios to standardize a way to evaluate trials and trial sites to see if the representation was adequate, as we said, compared to the population that we were treating.
Well, and it's such an important point, Orly and Wayne. I want you to comment on this because we have this archaic approach to clinical trials. We identify sites basically on historical performance. Maybe they performed well in a different disease state, and so we gather these sites, and then the sites are recruiting patients, through scanning of charts or through subspecialty clinics, heart failure clinics, and things like that. So there's enormous biases in who's getting into the trial. People who live in rural communities, people who live without transportation, people who don't have access to the subspecialty clinics are not being identified. And so already we have these types of barriers, as we were talking earlier, how artificial intelligence may help us take away some of those biases in identifying patients. But Wayne, you've published on this. Orly, you've been thinking about this a lot. How do we get better representation in our clinical trials at the site level?
Might ask Orly to speak first and I'll chime in afterwards.
Sure. I think one of the points that you raised is crucial, which is we're always seeing the same types of, or the same even sites in various clinical trials. And those sites tend to be large academic centers, which may not reflect the population at large, and certainly not a rural population. So it's really important to try to engage sites at different locations that are not necessarily tied to an academic institution, but are potentially standalone sites that have that serve a diverse or representative population around them to be able to draw from, and that will hopefully increase the representativeness overall. But I think one of the key ways to do this is to expand the types of sites that we accept into clinical trials as performance sites.
Yeah,
It's so important, that topic, and is one of the reasons, that Wayne and I came to Inova, because we said this is, outside the traditional academic walls, but there's enormous population that has been underserved by not having the clinical trial infrastructure in place in cardiology. Wayne?
Yeah, Chris, this is such a critical topic and a really important point. So excited to see Christian, Orlo, Mona, the Heart Valve Collaboratory, and others working on this, because I think we can do a lot better. I think we've been sort of trying the same experiment over the last 20 years and not really moving the needle. And what I mean by that is, instead of having a really sort of scientific approach to solving this problem, we've sort of relied on a lot of sort of typical speech. We've got to enroll more. Let's talk to these patients. Well, if you're going to sites that don't have access to those patients, you're just not going to get those patients enrolled. And I think what Christian and Orly have stated is quite relevant in that regard. There are about 17 different things we can do to improve clinical trial participation in terms of enhancing underrepresented groups. The National Academy of Science has a very nice paper on this. But one of the levers that we have not pulled is exactly what Christian had referred to, which is taking advantage of the natural heterogeneity of our sites and understanding where to choose sites to participate in clinical trials. If we're doing a study that has to have a large number of Hispanic participants, or African American patients, or other groups, we have to go where those patients actually live, and to institutions that treat them. And some of these may be urban, some of them may be clustered more in the Southeast, especially for Black populations in the United States. And we can't exclude the western part of the country, especially the Southwest, to ensure that we have access to the Hispanic population. And I love the way that Christian and you all are all starting to talk about this, which is look at, applying quantitative methods to it that fit nicely within our umbrella of regulatory science, participant to prevalence ratios, and understanding how the site selection and other levers that we can pull can move that PPR in the right direction, so that we're getting good representation, PPRs of over 0.8, for example, for all of our key subgroups. I think we can do a lot better, and I'm quite optimistic that you're going to see some changes over the next 5 years that will move the needle significantly in this realm. I think we're finally figuring it out a little bit better than we did.
Well stated, Wayne, and I think that's really going to be one of the emphasis is to use science, quantitative information to move this field forward. Dr. Fiuzat, what are your last thoughts on this topic as we wrap this up?
Yeah, I absolutely agree with everything that's been said and so nicely stated. I think I love the concept of the PPR, participant to prevalence ratio, understanding where those patients are, and then also maybe looking a little deeper at, is there a biologic reason that we need to have these, investigations? Is there a genetic difference in the people being treated, or the way they handle a drug or device? The way Wayne outlined in the beginning, are there bigger risks for certain groups? And so I think really bringing the science, emphasizing the scientific piece, and understanding the biology is a really important aspect here as well.
Well, thank you very much, my esteemed colleagues and panelists. As always with these podcasts of the Collaboratory, we identify a lot of issues. We don't always solve all the answers, but we're moving the needle forward. So I'm particularly pleased with the Heart of the Matter Heart Failure Collaboratory effort in this space to how can we do better in making sure our clinical trials are representative of the patient population that we are studying, that we're looking at these different therapies. And you've brought some significant light to this important problem. So I want to thank you and thank our audience for being here this afternoon. Have a great day.